Thank you. 'Tis the season for nonprofits to mention the other parts of raising money, which those of you that are members in the audience, which is a lot of you, yes? Nice. Thank you. You make up about half of our general operating budget, and the other half comes in the form of donations, and it comes between now and the end of the year. So those of you that are able to make donations, that is what keeps everything moving at Long Now and enables some of the amazing projects as well as the seminar series. And so we really thank you for that. And thank you for all your support over the last 20 years. Thank you.
DARPA is something I've been revering, admiring, paying attention to since 1972 when I first learned how important what was called ARPA then was to the development of computer capabilities and to indeed the Arpanet and to so much else. And at that same time, that was when I was paying attention to Buckminster Fuller, who said when you want to manage change in civilization, trying to change human nature is a big waste of time. It doesn't change much. But a point of high leverage is tools and the design of tools and the framing of the design of tools. And this is where DARPA, I think, has been especially valuable. Governments can do regulations, governments can do agreements, governments can do various things, but that the government would so adroitly push ahead the most blue sky and sophisticated approach to tool invention and innovation has been at the core of a pretty good society we've got going, I think. And now that we're entering what is being called a biological century, a biotech century, and genome editing is becoming another form of programming, DARPA is right in the thick, and the person who is in the thick of that is our speaker tonight, Renee Wegrzyn.
Good evening. Thank you, everyone, for having me. I want to thank Stuart and the Long Now Foundation for inviting me. When Stuart asked me to give a talk on engineered gene safety, it was a surprise. I didn't really think about coming to the West Coast to talk about what we're doing back in DC about safety. But really through conversations with Stuart, I came to realize that we share this enthusiasm for gene editing and how this can really have an impact on all of our lives across many different scales. From the scale of the laboratory benchtop, maybe at a university like MIT, where there's sophisticated researchers working together, to a benchtop that could be my kitchen or my garage. These tools are being democratized. How do we think of safety in those different types of settings, to whole ecosystems, to personalized medicine in an individual? I really started to feel this sense of urgency that somebody needed to do something about this and think about, hey, who has the plan for developing the tools that we can move forward safely? At the time that I started to think about this, I was an advisor to all of the ARPAs, to DARPA, IARPA, which is the intelligence community version of the Defense Advanced Projects Agency, of ARPA-E, this is the energy variation of DARPA, and they all were working on synthetic biology tools to forward their individual missions. And I started to talk to the program managers that I was working with, like, hey, do you think we can put a little bit of money aside and think about safety in the context of these projects? And was told that was a really good idea, but we have limited budgets. We don't really need to include that from the beginning. So I took it upon myself to go to the director of DARPA and say, hey, look, we're pushing these technologies out there into the world, really, and we're not accompanying them with the safety measures that should be in place. And the director at the time, Arthi Prabhakar, said, well, what are you going to do about it? And so I laid out a plan to establish a path forward that I'll share with you today. It's open for feedback. That's why I really like talking about this project, getting new ideas of how we can see where our blind spots are and try to fill those in. I like the idea of the Long Now Foundation, thinking about where the direction of a technology may go. And part of my job is thinking, where could something possibly go wrong? And what does this technology look like 5 years from now, 10 years from now? And having done many of these types of activities, even starting 5, 6 years ago, I'll tell you I completely missed CRISPR-Cas9. That's really what changed everything. Some of the projections, it's almost embarrassing to look back at what we were saying, what biology would be doing today, because we're really achieving science fiction level type activities. And I'll talk a little bit about what those surprises look like going forward. And if you'll just humor me for a moment, I'd like to tell you about the agency itself. This year is our 60th birthday, and DARPA itself was launched in response to a launch. This was the launch of Sputnik 1, launched by the Soviet Union in late 1957, and this was really not only a demonstration of incredible technological prowess, but also of military dominance. Now, here's a satellite that went into orbit and is now circling the Earth and passing over the United States several times a day, where the Soviet Union can have a look on what's happening in the US. President Eisenhower at the time put out this memo shortly thereafter to say we need to establish an agency, the DARPA, to have the singular mission of creating breakthrough technologies for national security. We can't let this happen again. We have to be the first and we have to be the best moving forward to keep our military dominance. And so there's many technologies that DARPA has developed throughout the years. This would be a talk that could take days if we went through them. We heard about ceramics earlier, ceramic bearings, but here's a few other technologies that you may be familiar with. So for the Apollo missions, DARPA was a critical early investor in the technologies that enabled the booster rockets to get to space. In the middle here, we have the Arpanet, and this was really a napkin drawing of the 4 original nodes. It's kind of blurry, but if you look, you can see at the bottom circle is UCLA, which goes straight up to SRI. And last night at 10:30 p.m. was the 48th anniversary of that first time that signal traveled the 350 miles, so the first internet signal. The program manager at the time, Licklider, who brought this vision of this program forward, he also thought about things like cloud computing, and we're at an age now where 140 characters reaches the ends of the earth within seconds, also with national security implications. Moving beyond that, this is the picture of the car in the top right corner is Stanley. This is the winner of the Urban Challenge. So precision guidance, autonomous guidance. And then finally, really the birth of some of the biological technologies at DARPA. Our flagship programs include revolutionizing prosthetics. We're now closing the loop where a man and machine can be integrated together. In this case, neuronal signals travel from the brain to a hand to actuate a movement, in this case, pick up a grape, but also have haptic sensation programmed inside that prosthetic to sense how tight you're squeezing that grape so you don't crush it, and that signal is relayed back to the user. Really incredible neurotechnologies. We're trying to outpace infectious disease, and where I sit is sort of at the interface between outpacing infectious disease and accelerating synthetic biology. So the diversity of the programs at any moment at DARPA is really a reflection of the program managers. It's an interesting organization where it's not top-down. Our director doesn't tell us what projects we should work on. We come with our idea to the director and say, I want to be a program manager because I believe in this vision and I want to make it happen. We're there for only 2 to 5 years. It's a very short tenure. With the idea, any one individual only has so many good ideas, and then we cycle out and new folks come in. So if you look at the DARPA website, really the array of programs we have are from that program management level. And so how do we make this happen? With this incredible sense of urgency, we walk in the door. We really have a very basic way of how we think about projects, and this is useful, I think, in well beyond DARPA. I'm going to use this after I leave DARPA. The Heilmeyer Catechism. So Heilmeyer used to be a director of DARPA, and he laid out, if you can't answer these 8 questions, then it's really not a project worth pursuing. It's what are you trying to do? Articulate it without any jargon. What's important here is that, for example, the director of DARPA, he is a hypersonics expert. He has no idea what I'm talking about when we talk about gene editing, so I have to be able to relay, this is a critical, urgent issue for national security, and here's why we need to address it. How is it done today? How are we going to change the state of the art? And who's going to care if we're successful? What difference will it make? What are the risks? These are not only technical risks, we must address those up front, but what are the ethical risks? What are the financial risks that we'll address? How much will it cost? How long will it take? And the way that we relate the urgency down to the folks that we provide funds to is what are the midterm and final exams to check for success. Every project that we fund is, we have 6-month, 1-year, 2-year milestones that must be met. If they're not met, then we stop funding and we focus our resources elsewhere to other teams that may be making better progress. I think that's an important point to point out. DARPA does not have these fabulous, shiny labs. We are an office building. We are people sitting behind a desk delegating funds out to people. We have a vision of what we want to make happen, and we look for the people to help us do that. And it's my understanding there's actually several people that I fund on programs here in the audience today. So I know you feel some of this urgency and pain. And so really, through the answering of these 8 Heilmeyer criteria, we seek to create and prevent that technological surprise. So if we think of genetic engineering and genome editing, what do the seeds of surprise look like? For people working in the field of genetics, these will be familiar images to you. Developing the tools to read and write DNA. So we've gone from the low price of a human genome for $3 billion, which is the entire DARPA budget, down to about $1,000 in 1 day to sequence a genome. Illumina and others are pursuing that $100 genome going forward. The ability to write DNA is also dramatically decreasing in price. And of course, the tools to edit genomes. So we've had tools to edit genomes for quite a while, but CRISPR-Cas9 gives us the ability to do so in a programmable way, really with just these 2 components where you have an enzyme that can bind to the DNA using a guide sequence that says, hey, this is your cellular GPS that tells you where you need to go inside the cell, make your cut here, make your edit here. DARPA investment was critical under each one of these technologies to bring these forward. And we are also critical in bringing many of these out and democratizing them by making the hardware to make DNA synthesis and sequencing and even editing out to the masses. Here we have the MinION sequencer. This is about the size of an iPhone, USB powered. The DNA write, this is the digital to biological converter. It's not quite at a price point where you can have it at home, but certainly at your research lab. And the idea here is to take genetic information from a computer and have a biopolymer printed out, whether it's DNA, RNA, or polypeptide, really bringing this, if it's in the laboratory setting, we're taking it out of a centralized component where we used to have some regulatory oversight. There is guidance for the providers of double-stranded DNA that they look at every order that's presented, and you can look and say, actually, this is Ebola sequence. You want to do that next level of checking to see who's ordering this. Are they allowed to be ordering this? Is there something suspicious here? There's not formal regulation. It's really a guidance. About 80% of the double-stranded DNA providers adhere to this guidance and say, okay, we'll be good citizens. We'll make sure that people are ordering things that they should be. And if not, they'll flag it for the FBI for further follow-up. Moving this technology outside of that centralized setting, I think is empowering. We can actually bring innovation to more amateur users. There could be things that we wouldn't otherwise get from maybe the group think of a university setting. But of course, this increases some national security risks in these components. And then finally, genome editing. In this example here, this is a kit that's advertised on the internet. Actually, I think it's developed here in the Bay Area, where the audience is really focused on DIY communities, so do-it-yourself biologists. But there's many sources that you can get CRISPR-Cas9, AdGene, and others. So these key enablers are really pushing this technology out there. I hope you're starting to think about safety and why we might need that. So just a little bit of background on genome editing. I think this is helpful for those that aren't really working on these tools every day. So this is a naturally occurring system. You would not believe how many billions of copies of this are in this room right now. They're part of our microbiomes and our microbes. If you imagine the market cap on this and how much you're worth, it's really kind of fun to think about and do that math. But the way these systems develop it's a way for bacteria to have an immune system. So they don't have antibodies and white blood cells to help them defend against infection. But what they do have is CRISPR-Cas systems. So a virus here, this little guy in the top corner, inserts his DNA into the bacteria to attack it. And that bacteria can cut up that viral DNA, make a copy of it, and insert that almost in like a library into its own genome as a memory of that infection, so that its offspring, the next time they're infected with a virus, it'll say, okay, I have a copy of this virus, I've seen it before, I'm going to cut it up and neutralize it. So this is the defense. And what was the big, really, innovation about 5 years ago, Jennifer Doudna, Feng Zhang, and others recognized that instead of using this viral DNA, what you could do is program any piece of DNA that you want as a guide to take this editor to a place that you'd like to in the genome. And using editing parlance, you could delete a gene, or you can copy-paste and insert a new gene. This is an incredibly powerful technology. So let's start to think about what does the surprise look like? What's the Sputnik of this type of work? So thinking of a bacterial system, what could we maybe program in mass into a bacteria? Enter George Church. And of course, he has taken every single picture in this moving image and mapped a genome sequence to it, uploaded that sequence in the rapid upload time of about 3 days into a bacteria. And this is on a loop. It's a very short segment, but each and every one of those pixels has been programmed into a DNA, and your readout from that system is sequencing that bacteria. So this is starting to show you what these technologies may enable one day. Is it DNA data storage? Well, the price of synthesis isn't yet at a price point that this is realistic in the next year or two, but maybe in the next 10 or 20 years it is realistic. There's a window of opportunity here that we can start to build in encryption systems, ways that we can start to protect this information if we want to, as a nation, use these technologies. Beyond that, it turns out that CRISPR-Cas9 is almost like a one-size-fits-all editor. So the rule as of about 5 years ago was if you wanted to work with a model organism, you could work with yeast and E. coli. There's great genome engineering tools, but to use another species, you'd have to create an entirely new toolkit. This is going to be millions of dollars of development. And I don't think CRISPR-Cas has met an organism yet that it can't edit. So from salamanders to pigs to rodents to bacteria, I'd love to, in the Q&A, if somebody of an organism, let me know. But so when you start to think about what we can edit now, we can think of editing these things as systems, as maybe entire ecosystems. Maybe creating new ecosystems of the future, or reviving ecosystems from the past, learning from them. There's folks in this room that are interested in de-extinction, bringing back things like the woolly mammoth out of the context of their original ecosystem. Maybe it makes sense to not only bring back the woolly mammoth, but the plant life that woolly mammoth may have enjoyed. Passenger pigeon, I know, is also of interest. This second image here is of a otherwise extinct plant, an image from the Harvard University Herbarium. So a creative director at a metabolic engineering company called Ginkgo Bioworks is looking at extinct species of plants in Harvard's collection to see, are there new enzymes and pathways that they can get from these extinct organisms that they can bring into the laboratory and start to generate molecules that are of value. And DARPA has tremendous funding on the Living Foundries program through companies like Zymergen and Amyris and the MIT Broad Institute to do just that. We've given them the task of generating 1,000 completely unique molecules that have not been made by biology before as their milestone and criteria going forward. So thinking about de-extinction, there is a time sometimes as a society that we decide to eliminate something from our environment. Smallpox is one example of something that we have eliminated because of the incredible suffering and death that brought to individuals. Countries still maintain stockpiles for fear of resurgence. And you may have heard recently that Canadian researchers actually reconstituted an extinct pox virus. In this case, it was a horse pox virus, which had also been eradicated as an agricultural pest. It had not been detected anywhere in the wild since the 1980s. And so as a society, collectively, we have eradicated these species of virus, and one individual or small group of individuals has made the decision to bring that pox virus back. So I want to emphasize that the power of small groups of individuals here using these technologies is immense, and it's important that we get the word out, thinking about, I'm not sure if somebody said in this context, what could possibly go wrong if we bring back a horsepox virus? And it's funny, but it's not funny. How do we address this going forward? And again, emphasizing, thinking about the power of one, genome editors can be applied to even bias the outcomes of inheritance in something that's called a gene drive. What a gene drive does is it couples a trait of interest with a genome editor. In this case, if you want to suppress something like a mosquito that's a carrier of vector-borne diseases like malaria or dengue, you can couple a trait that makes a mosquito resistant to malaria with CRISPR-Cas. And so through reproduction, it makes a copy of itself into the sister chromosome, so where mom and dad each would have a different copy. Now dad's copy gets copied into mom's copy, and there's 100% inheritance of your trait of interest. So for something like vector-borne disease, you may want to use a tool like this, and people have heralded, okay, we now have the technology to destroy all Zika mosquitoes, or name your favorite vector-borne disease. But at the same time, we have now created for the first time a tool that can lead to extinction. So under what circumstances do we decide to use this tool and understand how this might work in the environment? And is there a way to do this in a measured way that we maybe can do a local extinction and not a global extinction? These were some of the questions that I brought to the community to ask, how can we move forward safely in this world? And then finally, of course, human uses of these technologies. There's a lot of people are afraid to use the C word, the cure word for cancers here, but it really looks like an application of these technologies through some T-cell therapies and otherwise can reverse cancer even at very late stages. Industry that makes transgenic organisms for research tools use these same exact reagents to cause disease in animals. So where for those of you that worked in a mouse house in your graduate research, you may remember, years sometimes of making a transgenic mouse to do your studies, and that's just when you can start to then look at your drug of interest, et cetera. So CRISPR-Cas9 has made this a very precise way to introduce mutations. As new diseases emerge, it's relatively straightforward to put this in a mouse. And I don't think you need to be a biosecurity expert to recognize that there is a need for scrutiny when you look at a tool that can both cure and cause a disease. And enter industry. If there's human therapeutics that are at play, there's going to be a number of companies popping up to make the most of that technology. I could name any number of companies. Here's just a handful together. Their market cap is billions of dollars and growing. And what does safety, when we think about biotechnology and becoming a part of the bioeconomy, where does safety play a role here? And I'd like to just point out that many of these companies were built on the original prototypes of CRISPR-Cas9 to the promise that, okay, we can make these editors better and more precise. So when we talk about precision genome editing, I want to make it clear that these tools, all they do is CRISPR and the guide RNA come to the DNA and they make a cut, and that is it. They don't edit anything. They hand it over to the cell at that point, and the cell has DNA repair processes called non-homologous end joining or homology-directed repair, and they make the edit. So delete or copy paste, and I think you can hardly see it in this diagram, but there's a little green bar on one of the chromosomes, which is your on-target edit, and the red bars are off-target edits. So these tools, they're actually precision DNA damagers. This is another guide, same tool, where I think you can start to see the red coming up in this image, where often your off-target effect may outweigh your on-target effect. So how do we make these editors better so that we can start to direct the outcomes of repair to get the true edits that we want? There was a study published using these tools that it was kind of a very quickly published piece of work that said, hey, if you use these tools in mammalian systems, they cause hundreds of off-target effects. In that one day that this paper was published, there was $175 million loss in market cap from these companies. Very quickly, the companies and academia came to say, actually, if you look at the experimental design, it's probably overestimated the actual numbers of off-target effects. But still, the ability of an issue like safety to move a market like this, if the existential concern doesn't get you, then maybe the financial one will. And then people are taking this into their own hands. Folks that can't wait for the long timelines of an FDA approval. Maybe you have an inherited genetic disease and your clock is ticking. What can you do? And this is really, I think, a nice image from MIT Technology Review about an individual, in this case, who really well-educated looked, and he didn't have a disease per se, but he wanted to look at a way to increase his stamina and longevity. So he looked at the sequence for human growth hormone releasing hormone. He ordered a plasmid, that's what this circular piece of DNA is, that encoded this protein, this gene. It was delivered to him. He found a doctor who injected him. First, they did a tattoo, so you can see where he injected it, so if anything went wrong, you'd know where to cut out. And he injected himself there, but what's important to know is that when you inject yourself with circular DNA, it doesn't just get taken up into cells. You actually have to apply an electrical current to make sure that it gets in. So this doctor also applied an electrical shock to make sure that this was taken up. And I guess he's fine. I don't know what's happening, but I think that the real emphasis is this is one story of many that are starting to accumulate, that people are taking this into their own hands. If they can't wait for a therapy, they're going to try to make these therapies themselves. This individual argues that this does not pose any harm to the public, so it's fine to do. But I guess if I had the opportunity to talk to that individual, I would ask, well, have you talked to your insurance company about this? If something bad should happen, what does this do to the industry as a whole? We're trying to develop beneficent genome editing technologies, and if something goes wrong, what if it's misinterpreted as this field being a problem? And of course, we know the case with AAV therapies, where we for a very long time stopped doing gene therapies because of an adverse event. And we thought that genome editing was on the horizon. It's already here. Researchers in the US and globally are editing in the germline. So the Washington Post, I think, sums this up really well, saying new genome editing tools could cure disease or customize kids or aid bioterrorism. You pick. What I'm looking at DARPA is thinking about, okay, I'm starting to get really worried about bioerror. So this would be somebody, a well-intending individual, maybe the gentleman that's doing DIY therapies, causing an event that we had not anticipated. How do we mitigate that risk? What about releasing a mosquito, a gene-drive mosquito, into the environment? Creates a new ecological niche that gets filled by an even more deadly virus-carrying insect. How do we mitigate that risk of bioerror? And then I'll be checking the box for national security, and while I develop the tools to protect against bioerror, that same toolkit can be applied really to bioterror. They end with the same result. There's some lessons that I think we can learn going back to the Arpanet and the 48th anniversary. No criticism of the folks that brought the internet to us, but if they had the opportunity to look back and say, could we have developed security of those systems from the onset, where we wouldn't be worried about maybe cybersecurity as much as we are now. There's entire floors at DARPA dedicated to this. The NSA is dedicated to this. Would we be in a better position if we had built in safety from the beginning? So with this in mind, I went to the community and I said, if we want to use genome editing technologies, what are ways that we can control these editors, counter them if they should go rogue, and if they should introduce an edit into an environment or an organism that we didn't want to be there, how could we remediate that change and bring it back to baseline? And so that formed the basis for a program that I call Safe Genes, and the call went to proposers to address just these things. The first, the control of genome editing. This is turning an editor on where you want them, when you want them, and once they're done doing their job, converting its substrate, remove them from the system. It's when they hang around in cells after they've turned over their substrate that they start to accumulate those off-target effects. If we're going into the clinic with a gene editor, we don't have any antidote. We don't have any way to shut those systems down. if you think about taking a medicine that you may have a side effect from, if there is a side effect, you can stop taking that medicine, your body metabolizes it, and you'll go back to baseline. If something is permanently changing your genome and something goes wrong, you really want to have something at the ready with you to be able to shut down that editor to halt further damage. And if there is any damage inferred, then we can also try to remediate that damage. And the teams that we funded, we picked 7 teams. I'll go into the teams that answer the call to this, but just to give you a flavor of the different types of efforts that we're funding, we're about 6 months in, and we have folks looking at controllable genome editors. This is having a genome editor work only when you add a small molecule of interest. We have generation counters, so they're not quite at the level of 1,000 or 10,000 year clock, but they can count that they've gone through 5 generations, stop the gene drive, 10 generations, stop the gene drive. So these are smart systems. If we build these autonomous systems that we release into the wild, we want to build in fail-safes for our cars that are autonomous. Let's build them for our gene drives that are also trying to be autonomous. For countermeasures and prophylaxis, we have a broad swath of technologies we're funding now, from nucleic acids to small molecule inhibitors to protein inhibitors. Interestingly, most of these protein inhibitors comes from the phage themselves that were initially trying to infect those bacteria. Of course, they've evolved resistances. So nature is going to combat and counter these gene editors. And then finally, genetic remediation. This is an area that I would love to solicit new ideas in. So it's very difficult to keep pace with something like a gene drive. So something that biases inheritance to 100%. I can't tell you how many ideas that we got from individuals that said, okay, if you release a gene drive, let's create a reversal drive to reverse that same gene drive. Well, from a regulatory perspective, talking with the FDA, they kind of scratched their heads and said, okay, so you send out the first gene drive and it breaks, and then you want me to let you release the second one to fix it? Of course, that won't work in the long run. I think this is one area that we really need to think about. How can we develop technologies to keep pace with remediation and gene drives? And then finally, much of this work to date really has just been computer models. What could happen if we release a gene drive? What could happen if we use this therapeutically? And we're starting to actually do this testing in laboratory systems over many generations to see what are the impacts of evolution, what is the stability of these systems. To walk you through one example, for vector control, the state of the art, so now I'm going to the Heilmeier criteria, you can see. The current state of the art here is environmental modification. This is removing the habitat for mosquitoes to live in. It's spraying with insecticides, and more recently, it's sterile insect techniques. So you could irradiate a mosquito to make it sterile. These mosquitoes tend not to be very fit. Or you could genetically engineer a mosquito to be sterile. Groups like Oxitec are releasing mosquitoes. That's not a gene drive, but it's a sterile insect that, when released, it survives for about 2 weeks. And is sterile, so there's no offspring. The paradigm shift here is the gene drive, of course. So this would theoretically be a way to release one or a few organisms to suppress entire populations. If we decide to use a tool like this that could eliminate mosquitoes that carry disease, but also potentially make that species extinct, let's think about what development of safe genes tools would do to engineer the mosquito, drive this forward. With the tools afforded by safe genes, we can build in that control, keep it local. We can halt the spread of an unwanted gene drive if it gets beyond a geographic area that we'd like to confine it to. And we can also restore a system back to baseline, making the smallest change possible. We don't want to have to follow back in with pesticides or something like this. Let's restore that system back. These are the teams that we're funding. We have really a broad swath of academic researchers mostly, looking first at high throughput models. They give very short timelines for a DARPA program. Safety is only a 4-year program, so there's only a very limited number of generations that we can have for something like a gene drive. So to look at a greater number of generations, we're including high throughput models. So these are fly flies, yeast, and worms, and really using these really awesome liquid handling robots that allow continuous evolution of these systems to look at the different drive constructs to see which ones are the most robust over multiple generations, which ones do we have the best control over, and what are the components of evolution that really work to create resistances in these systems. We have 4 mosquito vectors that we're looking at that carry any number of diseases. And then finally, if we're successful, we'll create the first mammalian-based gene drive in a rodent. These rodents are invasive species in many island type of environments, but they also carry some diseases like Lyme and plague. And this last point, I'd really, people are kind of scratching their heads like, why does the DoD care about invasive species? And biodiversity of any given environment actually protects the organisms in that environment from disease. Having that incredible biodiversity is really helpful on the national security perspective because less disease means more stable nations. Here's just another tool in the toolbox that we can start to look at. There's multiple design drives, gene drive designs. We can think about threshold independent. This means small release, large impact, or threshold dependent. These would be where you have to release many species to reach a given threshold. What these designs afford is if a wind sweeps in and brings your mosquito to the next valley, that it won't reinitiate a gene drive there. It would keep it local. And then on the therapeutic side, there's 2 projects that we funded. One is from George Church's lab, and he has a very ambitious goal of trying to reverse damage induced by ionizing radiation. So if you think about it from the national security perspective, we are often first responders to a radiation event. You're, of course, familiar with Fukushima. In those cases, they sent in robots early on because they couldn't send individuals. Because of the damage that would be caused to those individuals, how can we find the cells that have been damaged with the ionizing radiation and restore them back to a baseline state? From the reagent delivery perspective, this is very difficult. I have 32 trillion cells. How will this team get to each of those cells that's been damaged? And so we'll see how George does. For Jennifer Doudna, we funded her to look at a really exciting application of RNA editors. So I've mostly talked about DNA editors to date, but this is looking at a virus. It could be an engineered virus. It could be a naturally occurring virus. In her case, she's looking at Zika and Ebola. And if we think about our current countermeasures against these types of viruses, the traditional targets are surface proteins. So our immune system sees the surface proteins on a virus and makes an antibody against it, and we can clear that out from our system. If you can have access to that entire genome, you can start to look at sequences that are conserved across many different strains of viruses, for the first time getting close to a universal neutralizing reagent, and that's really what Jennifer is trying to do. We talked about the digital to biological converter earlier. This is a way that you can start with only digital sequence of DNA that you know from a virus and create a countermeasure against it. And then finally, thinking about in the next 4 years of Safe Genes, we're going to be addressing some of these hard technical goals. These are the quantifiable. This is the data that leads to publications, that leads to new technologies and new companies, controllable gene editors, highly precise genome editing, inhibitors of genome editing, and gene drives that we can maybe one day field. And then also the soft questions. So what I've done in this program is made sure that we've set aside some funds for each team to have interactions with the regulatory bodies, with the community, with bioethicists to ask, what level of safety is acceptable? How do we appropriately monitor gene editing? And who does the decision making in these types of technologies? In a way, I'm also providing some soft governance here. There are no rules to the funding of gene drives. So in my call for proposals, I made up rules that said, okay, you have to have at least this stringent level of safety before we'll even consider you for a proposal. And this was done with conferring with many different experts, with the people that wrote the guidance on arthropod containment. What is this? Is this a BSL-3 level? But if we decide that it's safe, and do we just go from BSL-3 to open release? I mean, that doesn't feel right somehow. So really thinking about what does this look like going forward. We've also worked closely with the national academies, and these 2 reports are accessible on the National Academy's website for free. You can download Gene Drives on the Horizon, which really sets a path forward that all of our performers are following of how you can safely and in a staged manner go after these technologies. And then human genome editing, thinking about if this is somewhere that we want to go, how do we move forward safely? What do we think about germline editing? And what do we think about enhancement? And so that brings us to what's next. And should we really go there? So I like some of the work of the futurist Gerd Leonard, where he's made the statement that humanity will change more in the next 20 years than all of recent history. And I do think that we are on a trajectory towards this. On the downslope are some of the recent technologies that are making this possible. On the upslope, if you can see it, there's things like Human 2.0, human-machine convergence. These are things that are somewhere on the horizon that genome engineering and gene editing will be a part of. So how do we make sure that we can pursue this future in a safe manner? One possible trajectory, I'll tell you where we are right now with gene editing for humans. It's ex vivo cellular therapeutics. So this is taking cells out of an individual into the laboratory, editing them for some new function, whether it's engineering a T cell to attack cancer back in the body, but you can develop and QC all of those reagents in the laboratory before putting them back in the individual. The next near future is in vivo therapeutics. There's already FDA approvals to start to address some of these inherited genetic diseases from the eyes, liver, lungs, blood, skin, GI tract. You name it, in theory, there should be some solution that's afforded by genome editing that many will start to pursue. In each of these cases, this is taking an individual who is in a diminished, unhealthy state and bringing them back to a baseline healthy state. Do we go beyond that? What's next? What about enhancing that baseline state? So there's defensive enhancement, green guy with a shield. And so this individual, we do this already, actually. Think about vaccination. So when you're vaccinated, you actually are introduced to a change that is lifelong lasting that protects you from disease. This is an enhancement that most individuals are accepting of and even find it honorable and a must. We require, if our children go to school, that they must be vaccinated. There's other enhancements that would be maybe offensive, enhancements, performance, enhancement in cognitive state. Do we go there? What does that look like? What's the decision-making process to get us there? And then finally, an entirely new baseline state. So this is where germline editing comes in, and individuals in the near future, we already have demonstrated that it is possible, but what type of future does this look like, where there will be edited individuals, unedited individuals, who has access to those technologies? And this isn't something that just happens on its own. This enhancement as adaptation occurs through evolution, and I think this is a great graphic from this paper here, where they've identified genes in humans that where these are enhancements that make an individual well adapted to high altitude or cold temperatures or lactose metabolism. Anything that's not highlighted with a square are actual mutations in those genes that increase the activity. But those with the blue squares, these are just gene dose, meaning that if you just increase the level of expression of that gene, you can start to achieve better lactose metabolism, better adaptation to high altitude. We use drugs now to adjust to many of these types of scenarios, but in the future, will there be a question where we ask, what genes are you on? What are you enhanced with? Who am I talking to? And how do I know where do you stay in this level of enhancement? It's evolution really on steroids now in our control. And so one potential intermediate solution, if we don't want to go and actually change our genomes, is transient and reversible gene modulation for humans. So again, DNA editing, we're taking an editor, cutting the DNA. This is a permanent change. We could also modify the outputs of our genome at the RNA level. So the central dogma in biology is DNA makes RNA, makes protein. So can we interact at the RNA level in a transient way that once the editor is removed, that baseline level of expression is restored. There's an incredible paper last week from a group at the Broad Institute that actually is making edits at the RNA level. I work in a military organization, and somebody was like, everybody's excited about this paper. Can you explain it to me in a way that I can understand? And I'm like, okay, it's like jumping out of a plane without your parachute, and then someone intercepts you on the way down, put your parachute on, and you land safely. And it's like, here's a gene set loose, and you can correct it along the way so that when it lands, you have the healthy state of this end gene. So this is one way to transiently modify your gene's activity. But then, of course, for the aficionados in the room, there's CRISPR-A and CRISPR-I type approaches where you have an activator. You eliminate the ability of a gene editor to actually cut DNA, but it can still go and find its place in the genome where it needs to go, and you fuse that to something that is a transcriptional activator to turn on gene expression or repress gene expression. You can do so in the short term or also make changes that are long-lasting, even hundreds of days. So would this be a possible path forward? And if we want to do it, what does this look like? Well, what we can think about is in the context of druggable targets. So if we take a drug, we go after this gene network, we get our desired effect, but there's many off-target effects, and this is why there's toxicities and we're sick. But if you have a precision DNA modulator, like the one I've just described, you can really go in and just have your desired effect without those off-target chemically induced side effects. This might be something that's really an interesting path to pursue from the pharmaceutical perspective. In the near term, there's literature starting to accumulate on this topic. For the mitigation of Huntington's disease, this has been done at the modulation level of the gene and at the RNA level. For flu, so there hasn't been a lot of new insights to the flu lately, except in this case, a team overexpressed genes that altered the sugar structure on the surface of cells so that the flu could no longer invade cells. So they went from about 100% infection efficiency with influenza down to, I think it was 3% in the paper. This hasn't been done in vivo yet, but you're starting to get to a point where, maybe I don't want to knock down expression of receptors on my cell surface lifelong. This could have damaging effects. But if I know I'm going to be exposed to something, it's flu season, I'm getting on a plane, maybe I want to knock that down for the red-eye flight I have tonight. And then in the future, what about entire gene networks? We can start to multiplex several genes at once and knock out or knock in entire pathways. How are we going to do this? So this is something that technologies I'm interested in developing. Inherently, they're safer. They're not going to make permanent modifications to the DNA. But first, we need to identify what are those targets that we're going to modulate. If we want to protect against the next flu or the next infection, what are those targets? Getting to the tools to modulate those genes. Are they repressors? Are they activators? Is it a multiplexed approach? And then finally, formulations to deliver those tools. If I'm going to alter the cellular receptors on the surface, I want to make sure I'm doing that only in my lungs and not on my skin or somewhere where it's not going to be relevant. So the delivery formulation is really one of the most challenging aspects of biology right now, getting these tools where they need to go. And we're doing this in the context where we're talking with third panel experts and bioethicists, not only the folks at the national academies, but beyond, thinking about, okay, this is a defensive enhancement. How do we feel about that? How can we have conversations to understand if this is a place that we want to go? Acceptance for genome editing for somatic therapies and germline therapies is growing. Not quite there yet for germline and somatic enhancements. I think this sort of is grouping the offensive and defensive enhancements together, but really we're solidly on the defensive side. And just a note about where this is going. So just last week, the World Anti-Doping Agency banned all gene editing in sport. Not really qualifying what that means, but it's clear, okay, you could enhance EPO production. There's many things that you could do to enhance physical performance, and how are they going to monitor this? If you're increasing the dosage of a gene, I don't know if they'll be looking for the tool or what in these contexts. So I'll be curious to follow where that's going. And just to close out, we started with the Arpanet, and we'll end with a quote from Licklider, who was the program manager under the Arpanet. He had this vision at the beginning, understanding the impact that the Arpanet will have. And he said, to be online be a privilege or a right? And it's important to think about genome editing technologies as we change our world going forward. We don't want it to be a situation where there will be a select few that have access to these technologies. How do we push the conversation out to the greatest group possible to weigh in on where we want to go? With that, I thank you very much for your attention.
All right, deep breath.
I talk really fast, sorry.
That was fabulous. Thank you. Say a little bit about iGEM. We had a talk maybe 2 years ago from Drew Endy about the sort of iGEM revolution, and you've been involved in that as a judge, I think.
Mm-hmm. That's right.
Say what iGEM is and how you think it relates to what you've been talking about.
Sure. So iGEM is the International Genetically Engineered Machines Competition. I think it's been around for about 10 years now, if I'm not mistaken, more than that. And the idea is it's a summer lab project for students at the undergraduate, graduate level at universities to have an idea about synthetic biology and make it a reality over this short period of time. They have access to what are called biobricks. These are the building blocks, the genes that can be used to sort of start their designs, but they can build upon these designs. But I think one of the things I really like about iGEM is there two things actually. So one is each and every project has what's called human practices, and so this is thinking about safety in the context of those projects. So
when the team comes together, this is right at the beginning how they're approaching what they do? That's
right. Yep. They put out a plan to how they're going to address safety in these contexts, and they also engage with their local communities. And that's part of the entire project, and they get scored on that. Interestingly, iGEM, sometimes the safety requirements of the iGEM competition are more stringent than the requirements of the countries where the individuals are coming from to compete.
Where do they come from?
All over the world. I think that if there's a country, you name it, they're participating in iGEM. So that's actually, I think, been a great way to establish norms in synthetic biology and have those students take them back to their home countries.
Related to that is a question from Jen of, can you describe the international conversation in gene editing safety? So iGEM is clearly part of that. What's sort of the graduate, postgraduate version of it?
Sure. So I was actually just 2 weeks ago in Germany. The Volkswagen Stiftung funds conversations about many topics, including genome editing and security implications. So there was representatives from the Department of Defense, from the State Department, and elsewhere within from the United States, but from countries all over the world. They brought students and individuals together to talk about gene editing and what that means. And it was actually a really interesting conversation. I think it was the first conversation to get to know each other and get comfortable with how we think about these technologies. So some countries are pretty bullish using these technologies. GMOs are a great example, right? The US is like, go GMO. And then Europe is not. And although I did visit a research lab in Europe that was developing GMOs for other countries outside of Europe. So, and it's an interesting point, and it sort of boggled my mind. I'm like, how could you be developing GMO plants and you don't use them in Europe? And this person who relayed this to me said, we didn't have the conversation with the public at the time for developing the GMOs. So the researchers in Germany and elsewhere in Europe were pursuing these technologies, but those in the public didn't have a say, and so there's a dichotomy there now.
So how does the public have a say angle on this? And one of the people you're funding is Kevin Esfeld, I think, who's hard over for basically you're asking public permission before you even do the research and things like that. What's your perspective on that level of transparency and permission seeking?
So from the Safe Genes perspective, we want to be as transparent as possible. We've provided funding for each of our teams to make sure that they can have those conversations publicly. We want them to publish other research. We're requiring them to share their data. I do think that with that transparency, there's a responsibility to be there to translate the information that you provide to the public. So just a data dump of sequence on the public isn't going to do much good. But being able to confer with them and say this is what this information means and why it's important, and be there to answer questions, I think is going to be critical.
Well, surely there's some kind of a lag or something here. I mean, it's taken a while for people to get sort of, they went through the bits and bytes, the level of understanding what programming digitally is about. And genetics are way more complex and strange and personal than, some nice, safe machine across the room. Is there and it sounds like the many young teams that are involved in iGEM are sort of one way of them engaging this as practitioners, not just as somebody hearing about it and worrying about it. But there are a lot of people hearing about it and worrying about it, and for whom genetics has a past of the bad seed and these various things that we worry about genetics. Is there going to be just a lag of people saying, look, it's genetics, and I don't understand it, therefore don't do it? And then how do you overcome that? Because the understanding itself is difficult. And how deep do you have to go? Do you really have to understand the mechanism of CRISPR-Cas9?
Right.
Which I promote avidly, and frankly, I could not explain it.
Right. So, I don't have a perfect answer to your question, but I do think it is important to engage the communities, and I try to not be at that. We just have to educate them more so they understand, and I don't think that's really it's listening in both directions, and so we have a gentleman, Jason Delborn. So he's from NCSU, funded under safety, and so I'm just going to do a demo of what he does. Give me your hand, Stuart. So we're engaging now, and part of this engagement means that we can sway each other. So you may have an opinion, I have an opinion, but we're both movable in this conversation. And I love that demonstration that Jason has, because if you're going to engage with the public, do it in a way that's meaningful, that you're both vulnerable, and that there is some movement there. You may not completely understand each other or where you're coming from, but you're open to that movement.
Outstanding. Kevin Kelly, back on the international, asked, are there similar programs to DARPA in China and Asia and Europe, or are you guys unique?
Say it again?
Are there similar programs to DARPA elsewhere, like in Asia and Europe and Africa?
I just saw a news clip that I'm so embarrassing. I'm forgetting the name of the leader in France, Marie. Le Pen. There was some France just recently. I apologize for not knowing the name. Has stated we should get something like an ARPA or a DARPA agency.
In France.
So there are others, I think, that are looking at DARPA as a model. There's certainly companies that are thinking about this, but specific names, I think there are sort of counterparts in the UK. We have DSTL is an organization there that sort of models some of their work on DARPA, but there's not a identical clone somewhere elsewhere in the world.
Well, DARPA itself is a very American national government organization. Do you guys sort of have your own outreach to other countries? How does that work? Do you engage other countries in the research?
So we do. We have delegates from other countries come and visit DARPA to learn about the organization. And what we do, we sometimes do that at the office level. So I'm in the Biological Technologies office. So we've engaged with counterparts in the UK and Japan recently are some examples I can think of. And other offices do so as well. But of course, we are a national security agency, but we have allies across the world that we work with.
Yeah, I was just with Ryan in Australia at CSIRO, which is their government research organization. They got some of the money that you guys just now put
That's
out
right.
for
Yep.
research on, is it gene drive they're working on or something else?
Yep, so they're looking at some of the environmental assessment and risk assessment for the technologies, CSIRO, so this is with the mammalian gene drives. We also fund groups from the UK on some of the mosquito work, so a group at Imperial College and a group in the Purebright Institute.
I heard the amount that's going to this current sort of tranche of research is $65 million, is
That's
that
right.
More coming later, or how does that work?
No, well, not from DARPA. So the key of DARPA is we make that early investment to demonstrate that something is actually possible. And early on, we try to line up those advanced developers who can pick up our technologies, whether they're a company or another government entity, to develop that further.
A couple questions here relating to basically digital questions. Signu Udenberg, it looks like, says, speaking of designing safety at the beginning, Eric Schmidt gave an interview where he said if they thought about safety at the early stages of the internet, it would never have been developed. So is safety potentially a strangling thing that happens?
I personally don't think so. An analogy that I like to use is if you think about your car, you don't have your brakes in your car so you can go slow. You have the brakes so you can go very fast, get where you need to go. But if and when you should need to stop, you have the power to do so. And I think we need that personally for gene editing. And I do see this as a way to accelerate bringing these technologies to market.
I can imagine if it's done in a way which people really get, it also accelerates the sort of public permission, the social license to proceed with these things. People have some confidence that it is moving ahead safely. We'll see.
I think we've learned from biological systems too. If we build a system and optimize it and then try to retrofit safety into that system, it breaks the system. And so it's really critical to have safety as a design feature early on. There's a great paper by Jim Collins where he developed these kill switches. So he had this E. coli that's dividing, added a kill switch. It'll kill the bacteria really for the first generation or two. It works great. But if you monitor those systems over many days, evolution kind of, there's no greater pressure you can put on on a system than to kill it, right? So it's going to try to evolve around that. And so, if we can build that in from the beginning, would those systems look differently? I don't know, but we're trying to establish that as part of Safe Genes.
Another digital-related question from Chris, either A or H. Can you speak to the efforts, if any, to combine artificial intelligence, machine learning, and so on, with gene editing? Is that something which is enhancing all of the trait analysis, things like that? Is that helping?
So part of what I'm interested in pursuing in future efforts would be looking at if we want to identify gene targets that we could potentially modulate, there could be some information that we learn in terms of targeting those genes that AI could be supportive. And in the Living Foundries project that I mentioned, we do apply AI there through several of our teams to search the genetic space, sequence space that's out there to look for new enzymes that can catalyze reactions to create new molecules that are of interest. So it's applied today. I'm not sure the exact application that you mean here. So I think it's coming together. It's not quite there yet, but that will, I think, be an increasing part of it going forward.
The whole genetic universe is such a sort of data vast domain that one can imagine one would like every possible help you could get to be able to identify things you care about.
Sure. I think thinking about human gene editing too, when we think about AI, so full disclosure, I'm not an AI expert, so what I say right now may not be right. But what I understand is there's sort of, you ask the system a question, a little bit of a black box, you get an answer out at the back end, and it's a probabilistic answer. But how the AI system reaches that answer, we're not quite sure of. And DARPA now is trying to, we have a program called Explainable AI to say, okay, if we get to an answer.
Explainable AI?
Explainable AI. So how do we get to that answer? And most importantly, when the answer is wrong, how do we figure out how the system got it wrong? And if you think about that context for gene editing, if we want to use these tools, AI tools, to identify targets for disease, if we get something wrong and it causes maybe a worse state than the original disease that we're trying to correct, we want to be able to go back in that system and understand, what did we do wrong when we allowed the AI to design that system.
Explainable AI is pretty interesting because one of the things that's been emerging from some of these just sort of learning from the systems that are out there is that they develop algorithms that are so hidden in the code, we don't know what they are.
Right,
and
exactly.
This is, you're trying to make it so that we do know what they are.
Right, exactly. I'm not, somebody else is.
Well, that raises, so how many program managers are at DARPA right now?
It's a pretty lean organization. There's about 100 of us.
100?
Is that a lot?
And do you characters interrelate with each other? So if you have a potential, application of AI, is there an AI program manager you would say, would you look at this situation and help me out, or how does that work?
Absolutely. So, we, as everybody is, we have a full day, but we always try to talk with people on other floors. The way DARPA's organized is each floor is kind of a technical office. And so I do talk with folks in the I2O office, so Information Innovation Office, about using the tools of AI and actually even just a data commons for the safety community. This is where they're going to be putting their data, not only to share among the safety teams, but to have other people working as modelers in areas that maybe have nothing to do with biology to look at the data set and say, are there things that we can learn from the data that's being accumulated over many generations, that there may be some unique insight. So I think a lot of the really exciting innovation at DARPA comes when multiple disciplines come together and do great things.
Hot stuff. Clayton asks, what is CRISPR's potential role in the development of proliferation of biological weapons? And is it already too late? Or, National Security, DARPA, Defense Advanced Research Projects Agency, presumably the worry about bioweapons is got to be in there. Is CRISPR a potential significant tool for bioweapons?
So it is another tool in the toolkit, certainly. I gave you the example of the horsepox virus, so there's a real tangible example of some of these tools, not specifically CRISPR in that case, but new genome tools making those possible. So I think that, yes, it will play a role. It's going to be pushing these technologies out. I hate to speculate. I always get really uncomfortable speculating. I don't want to speculate, but I think this is a place where you can look to science fiction and some of the books that have been actually been published recently on the topic come forward with really frightening scenarios about where you could do things like I could commit a crime, but I could use CRISPR to put my CODIS, my STR. What the FBI uses is just kind of snips to say, okay, here's these repeats that are unique to Renee. I can CRISPR those onto you, so now you're responsible for the crime, Stuart. I don't know how near term that is, but these are some of the fantastical scenarios that may become a reality.
So part of this must be monitoring. Genes are very small, and they're hidden in a very thick bush of other things which are genes and not genes and so on. So somebody deployed a thing that one might consider a direct or indirect bioweapon, or one of the things that you care about did get, in a sense, loose in a way that was not intended. It was supposed to be self-limiting, and it's not self-limiting. Are there genetic level ways to detect that kind of problem?
I don't think those exist right now. So you could monitor for the tool, for the actual editor itself, but that's probably going to be short-lived. So if it's going to introduce an edit into your genome, it'll be there until it turns over that substrate, and then it'll be gone. So you have to catch it in that window of opportunity. There's another ARPA agency, so IARPA, that's developing a program specifically to look at, can you monitor and look for signatures of engineering, whether it's CRISPR-Cas or whatever the next editor coming down the pipeline is. Can we look at a sequence and know if it's been manipulated? But those don't exist today.
That's like the ones that are looking at photographs to tell if they've been manipulated and things like that.
Absolutely.
IARPA is intelligence ARPA, I assume. Of course, this question, there will always be a version of. Bob Kopek, it looks like, says, how long before some high school student creates a unicorn as a science fair project?
Oh, that's awesome. I'm sure that my daughter wants to be
probably a small unicorn.
That person.
My pony.
Well, we're doing the reverse. So I know companies are developing hornless cattle now. So maybe they need to reverse engineer those systems, or just remove one of the horns.
You know why hornless cattle, right? I mean, this is something we're evoking at Revive and Restore because, I'll say a little about it. Cattle hurt each other with their horns, and so the people who are caring for cattle dehorn them, and it's painful for the animal. There's a serious animal welfare issue here. And so it turns out there's a relatively simple gene edit which already occurs in some cattle. They don't have horns. Use that in other cattle that now they don't have horns, and they don't hurt each other. That's the hornless cattle thing that's coming on. It looks good. The cows look a little strange. They apparently get used to it, and they're not hurting each other. And the animal welfare people should be cheering, and I hope are. But it's the kind of thing that we're seeing here.
Right. Can I tell you a story since
Go
you brought
ahead.
up animal welfare? So DARPA just recently got a letter from PETA commending us, believe it or not, on a program called the Microphysiological Systems Program. So MPS, this is it's essentially an on-a-chip program, so organs on a chip, to recapitulate what an animal model might look like and behave like, so you can do all the experimentation on that chip and not actually have to sacrifice an animal. So PETA sent us a letter of commendation on that program.
PETA's interesting. PETA came out strong in terms of basically vat-grown meat and impossible burgers and things like that. And I agree. I think anything that is reducing animal suffering and is reducing animal agriculture impact in the landscape is a good thing. Wayne asks, what unexpected risks have developed in your current projects? And generally not just unexpected risks, but what are the surprises so far? It's research, so you're supposed to be surprised. What's turned up?
So something that's happened in my projects over the past 2 years is I used to hear from individuals, so where's your progress? And they'll say, well, we're cloning this right now, so we're busy in the lab making this happen. And that's changed to, well, we're waiting for the DNA to arrive. So nobody clones anything anymore. And I've been surprised at how quickly this has become the reality. Everybody orders their DNA from a third party.
Ed,
Kids these days.
say a little more about that, because there's sort of, these simplistic biobricks for microbes that iGEM was doing. It's gone beyond that, it sounds like. Or is it?
Right. Yeah. So people can, for the Living Foundries program is a great example. So we challenge the teams to create molecules of interest. Sometimes they have to stitch together, maybe 10, 20 enzymes. To catalyze a reaction across these multiple steps. And so they'll order each of those genes from a DNA synthesis company, and then they can assemble those in-house into these long stretches of DNA. And they don't really need to do the cloning as they used to do manually, amplify pieces of DNA, stitch them together. They can just order those pieces now. So that's
been expensive?
So from a funder's perspective, I do find it to be one of the most expensive components on our projects.
Is that one of those areas where
but in the grand scheme of things, yeah,
the costs are going down.
Is going down.
You mentioned molecules of interest. What are molecules of interest?
Okay, so sorry. So from the DoD perspective, these can be anything from a small molecule, a drug, to an adhesive, a coating, new materials, epoxies, hardeners. Really, you name it, and we want to be able to access that chemical space. So, as a country, actually, not just the DoD, there's many things that we source, either that are very costly because we may extract them from biological systems, or we have to source them internationally. They're not available in the United States, and so how do we bring those capabilities here that are cost-effective and produced domestically? I think there's a lot of Zymergen people in the house here, if I'm not mistaken, doing this as part of that project. And I'll say of the 1,000 molecules we want them, the 3 teams collectively to produce, they've made 551. So it's working.
And what makes them they're not randomly making molecules. They're making molecules that are somewhat targeted or somewhat adjustments to already known molecules, or what?
So there's 3, to get specific about how the contracts were set up, there's 3 levels. Some are, the first level of molecule are things that have been made by biology already, but making them better and more cost effective. So those are, you see the low hanging fruit. There's a pathway there. They know how to do it in theory, but make it better. The second category are things that there are enzymes that exist in nature that should theoretically be possible to make, but they need to be stitched together into a single organism to do that. And then there's the third category, which are just things that have never been made by biology before. And so these are very challenging to engineer. This is, we've had one performer recently, our grantees we call performers, say, I've run out of database.
That's pretty good.
Yeah.
Perform or go away.
Sort of. But yeah, so they look at really metagenomics, enzymes from the from deep thermal vents in the ocean to high peaks, to look at the enzymes that are present in these organisms to stitch them together to catalyze new reactions.
Wow, okay. Question on George Church's program, which I hadn't realized until you mentioned it, of basically being able to correct radiological damage to people. Since the major human impact of travel in space is the exposure to cosmic rays, which shorten everybody's life basically by doing a lot of DNA damage from the radiation, is one potential product of this, if it works, people that are comfortable traveling in space without a whole lot of cladding.
So I don't think that any of these technologies would replace shielding or protective equipment that would be required for a long-term space travel mission. But your point is well taken that we're pushing to travel to Mars or the moon or elsewhere, and I don't think we're keeping pace with the development of those, the hardware and technology. We're not keeping pace with the development of really protections for the human element that will be there. Are there ways that we can protect those future space travelers from radiation damage, reverse that radiation damage?
Protect from would be really nice. I raised a question with you earlier, which was one of the things going on, as you may know, in climate change and geoengineering is the desire to be able to capture carbon from the atmosphere, carbon dioxide from the atmosphere, and fix it in a way that it doesn't keep cycling back. And there are some organisms in the oceans which do that. Certain phytoplankton do it, for example. But they don't do it as much as we would like in order to get back down below 400 parts per million. So one could imagine engineering some microbes which are comfortable in the ocean and can proliferate widely, that are extra special good at basically collecting carbon dioxide and probably putting in some kind of shell or something like that. When the organism dies, the shell goes to the bottom and is geologically gone for a long time. Is that doable? Is that okay?
So I don't know. I'd love to see a project plan that looks at that. But if we think about
you could make it happen.
It could. So number 5 of the Heilmeyer Catechism. What are the risks? One of the things would be what if that system you just described continues in an uncontrolled way? So that we not only fix carbon, but then we start to sequester carbon. We move into the next ice age.
And they're in the ocean, so there's a lot of them out there.
Right. If you build in safety from the beginning, maybe it becomes viable.
Yeah. How would you build in for a system like that, which you're basically releasing into the wild? And most of these things we're talking about are ecosystem level, like rodents in the wild, like Lyme disease carrying white-footed mice in the wild, and so on. What are ways to localize and potentially reverse, say, gene drive applications of getting deleterious genes in the case of animals you're trying to reduce or extinct? What are the methods now of modulating that?
So I don't know that they exist yet. We're trying to establish those in part under the Safe Genes program. It really the best solutions I've seen so far are reversal drives. So if you drive a trait that becomes
which the FDA won't let you do.
Right.
You already fucked up. You don't get to fuck up again. I mean,
right. Exactly. So I don't know what can keep pace with that, and I welcome some concepts of how we can address that. But
well, you mentioned generation counting, that a thing might sort of only apply for so many generations, and then it either stops or poops out or something. How does that part work?
Sure. So some of those are self-eliminating, where you can use a genetic trick to flip a gene through every cycle of reproduction. That a promoter is turned on in the germline, it flips the gene, and you can insert any number of flips there. You can put 10 flips, and that would be a 10 generation counter, for example. So it's a physical and permanent change in that genome to maintain that state. This is one way you can do it. You could also have those systems become self-extracting, where you can have there's a multitude of gene editors. There's Cas, there's CPS, CPF, and you can have one target the other to self-eliminate. Would be another example.
So you've been at this how long now at DARPA?
Almost 2 years.
What have you learned so far? Kevin Kelly puts the question, what have you changed your mind about recently after working on this?
What has changed my mind about working on this? I definitely feel more confident that I was like, is anybody going to think this is a good idea? And the more I learn about the impacts that these technologies are having.
Say a little bit about what gave that I think I heard that you sort of went in with this proposal, and usually program managers have 2 or 3 proposals before they're taken seriously. But you were taken seriously right away.
Well, I'd been around a little while, so I sort of knew the DARPA way of thinking. And it was, genuinely, I really wanted to, this is going to sound cheesy, but I wanted to serve my country in this way. I never served in the...
Excuse me, that's not cheesy.
Okay. But I really felt, this is something that I wanted to do. I was excited at the innovation coming out of the United States, and I wanted to protect that so that we could pursue these technologies. So I was maybe one of the pleasant surprises is there's actually a lot of other people that want these technologies to move forward safely. And maybe that's not surprising as it should be. I think I've been surprised at finding common ground in conversations about conservation, for example, where maybe I hadn't in cosmic exploration. There's insights from new fields that I've learned about over the years that have just been really. I'm leaving myself open to those conversations, and hope to continue to do so. So as an individual, I've learned that having those open conversations has really been valuable to me.
Thank you for the open conversation today, and it's going to keep expanding. Thank you.
Thank you. Thanks, Stuart.